Hi guys, let's do a biochem lecture today. Let's talk about glycogen synthesis and we'll see how far we get. We might get into glycogenolysis as well. So this will be for, I guess we're in week 7 of fall of 2025. Off we go. One bird for you today. So I got this one, when was it? Wednesday? Last Wednesday. So super rare. I've never even seen one before. So this is a short-eared owl, and they're nocturnal, so they don't fly during the day. But it was cloudy, and I think he must have been really hungry. So surprise, surprise! Here comes a short-eared owl right in front of me. So very cool picture. All right, glycogen synthesis, also known as glycogenesis. So AKA glycogenesis, we just said that. Extremely important that we have constant glucose around all the time, right? Some cells have to have glucose. They can't live without glucose. Red blood cells are 100% reliant. They have no mitochondria, so there's no Krebs cycle or oxidative phosphorylation. The renal medulla is also 100% reliant on blood glucose. Brain cells are arguably 40 to 50% reliant on blood glucose. They can use ketone bodies during starvation, but they have to have at least some glucose around. So those three are called obligate glucose users, meaning they have to have blood glucose. And if you eat every 4 hours, 24 hours a day, no problem. They'll always have glucose. But of course, that doesn't happen. You have your last meal at, 6, 7 o'clock, and you wake up at 12 hours later, that blood glucose is long gone. Your dietary glucose lasts about 4 hours. So what are you going to do when that runs out? You have to make new glucose, or you have to release stored forms of glucose into the blood to keep these tissues alive right here. So that's why this is important stuff here. And these are all the tissues that need at least some glucose. We're just going to, you don't have to memorize all these, but you do have to know these three for sure. Those are the obligate glucose users. They have to have some around. Other ones that are very high are the cornea, the lens, Sartori cells in the testes. So those, they can use other things. They don't have to have all glucose, but they would prefer to have glucose. All right, so what's the trick? Well, the trick is to eat enough food so that there's extra glucose to store for a rainy day. And that rainy day will come during your overnight fast every night. And so, yeah, we have to have another method to put glucose into the blood between meals. And here are two such methods, and in order. Glycogenolysis. That is the breakdown of glycogen to do glucose molecules to release into the blood. And then there's gluconeogenesis. Gluconeogenesis, we'll do that one next week for sure. I'm probably not going to do, because we're starting to run out of time, probably won't do the glycolysis, the Krebs cycle, electron transport, because you probably had that a million times. These you probably haven't, so that's where I'm going to kind of focus on these things. So some fun facts. After dietary glucose, we said this is used up after about 4 hours. What happens in the pancreas? The pancreas releases a hormone called glucagon. Remember, when blood glucose is low, glucagon is high. When blood glucose is high, what's high? Glucagon is low, insulin is high. So glucagon will then signal the cells of the liver, which are called hepatocytes, to break down some of the stored glycogen. Glycogen is nothing more than a bunch of glucose monomers on these little branches, so we can rob the glycogen tree and steal the glucose off it and release it into the blood. Great. The liver glycogen glucose is then released via the process is called glycogenolysis. That will arguably last for about 24 hours. The books are all over the place. Some say 12, some say 18. Just an easy way to remember is 24 hours. We'll go with that. But on boards, if you see something that says it lasts for 12 or 18 hours, those could be right answers as well. If it says it lasts for 1 week, that's a false answer. If it lasts for a couple hours, that's a false answer. So we're going to go with 24 to make it simple. Then what happens after 24 hours, and we still need to get glucose into the blood, and we can't run glycogenolysis anymore because there's no more glycogen left. Then we have to go to the process of gluconeogenesis, which is a little more complicated, I think, than what we're going to talk about today. So before we talk about breaking down the glycogen, it's probably smart to talk about making it first, because if you don't store glucose for a rainy day as glycogen, then there's nothing to break down. So we're going to talk about glycogen synthesis first, also called again glycogenesis, glycogen synthesis. A note that skeletal muscle can also make and store glycogen. So skeletal muscle can run glycogen synthesis, but it can't share. When it's starving and needs glucose, it can break its own glycogen down for its own use, but it can't share. It doesn't have glucose 6-phosphatase is the problem, and therefore it can't share. All right, so let's look at glycogen synthesis first. So it occurs in the hepatocytes and the skeletal muscle, but we said that's not really, we can't share that, so I'm not going to say a whole bunch about that. We won't get into the mechanisms. We're going to focus on the hepatocytes because that's what can release it into the blood after our 4-hour meal goes away. So specifically occurs after the cells have produced enough ATP to satisfy their own needs. So what does that mean? So you're not going to, if you're hungry and you just start eating, you're not going to immediately start making glycogen. You're going to feed all the liver cells, or the liver cells are going to take that glucose and they're going to run cellular respiration to make ATP for their own needs, to power their own cellular processes. So maybe that'll take an hour or so. But after that, once all the cellular processes and the hepatocytes are all happy and they've been fed, we still have a couple hours of glucose coming in. So what are we going to do with that? Well, hepatocytes will take that and they'll make glycogen from it. See how that works? Great. How do we make it a little bit deeper? So here's a hepatocyte right here, a cartoon of it. And there's a key transporter that you need to know. So you just ate a bunch of food, and now the glucose is flowing through your blood. So you have a GLUT2 transporter. You do need to know that. You have a GLUT2 transporter that takes glucose out of the blood or out of the interstitium, because it goes from capillary into interstitium, and now it goes in through the cell down its concentration gradient, because you got lots of glucose out there and your cell has very little in here. It's starving for glucose, and therefore it just moves through GLUT2 transporter inside the cell. And now we would have a problem, because if we move glucose in here, pretty soon the amount of glucose here will be the same as the glucose out here in the interstitium. So what we need to do is trap it, and I've talked about this before. In hepatocytes or liver cells, we have hexokinase 4. So there's 4. I think there's really 6 hexokinases, but hexokinase 4 is also known as glucokinase, and we won't get into the energetics of that right now. It's probably getting a little too deep for a board review, but you do need to know that it's GLUT2 transporter on the cell membrane that lets glucose in, and the trapping enzyme is hexokinase 4, aka glucokinase. And what it does, it just sticks a phosphate on the glucose, slaps it on carbon number 6, and now we have a new molecule, a new look. We have glucose 6-phosphate. And so glucose 6-phosphate is different than glucose, so it keeps the concentration gradient alive. And glucose keeps moving down its concentration gradient into the cell because it's trapped by hexokinase 4. Cool? Got it? Great. And just while we're on this slide, do I have a bigger picture? Yes, I do. While we're on this slide, if this is the first food, like it's an hour after you fasted all night and you're eating your first food, the glucose 6-phosphate will run through glycolysis to make pyruvate, and then it runs through the Krebs cycle. Remember, the pyruvate has to move inside the mitochondria, and in the mitochondria it's converted to acetyl-CoA, and acetyl-CoA can combine with oxaloacetate to make citrate. I think that was on your last test, or was that? I forget where that was, but I just remember seeing a question on one of my tests like that. And there's the Krebs cycle, and the NADH and FADH goes into oxidative phosphorylation. The electron transport chain is the first part of oxidative phosphorylation, and the last part is the ATP synthase. That's the one that makes ATP. Definitely want to know that one molecule of glucose makes 32 ATP. But after tons of ATP builds up, and after a lot of citrate buildup, these two start inhibiting the glycolysis. And therefore, it's going to push, glucose 6-phosphate's not going to want to go this way. And it's going to want to go some other direction. And the direction we're talking about today is we're going to go into glycogen synthesis with that. It could go into the pentose phosphate system. It can go a lot of different directions. This is a kind of a hub right here. It can go different ways. But today we're going to push it into glycogen synthesis. So yeah, everything we said. Once the hepatocyte is basically made all the ATP, then we have extra glucose 6-phosphate to go a different direction. And here I drew it in. It's not going to go this way anymore because all these enzymes are blocked from high levels of ATP and high levels of citrate. And there's other regulators as well. Instead, it's going to go a different direction, and it's going to make glycogen. Okay, yep, specifically it's pushed into the glycogen synthesis pathway to make glycogen, and which can be used after the 4 hours has expired, and we've said that already. All right, just a word about glycogen. So this is a glycogen molecule. This is pretty accurate. On boards, they usually don't go this deep, but I put the glycogenin core in there, and I put the primer on there. So this is a real true glycogen. All these little tiny dots, those are all glucose molecules. So there's a lot of them. So some fun facts about glycogen. It's found in the liver and muscle. It's made of about 50,000 glucose monomers. So one molecule of glycogen contains 50,000 glucose monomers. And what's a monomer? Each one of those yellow dots is a monomer. It's just a glucose unit, you could call it. The monomers are usually, this drawing's a little bit off, but the monomers typically, the little glucose beads here, they don't typically go more than about 10 per branch. And then there's something called branching enzyme, which will create a new branch. And so we have a structure that started from a glycogenin core. Glycogenin builds a primer, which is this too many here. It's about 10 glucose units, and those are permanent. You can never get rid of those. And then the new glucose units are put on the primer, and after you get 10, they'll be cut and another one will be started. Another 10, it'll be cut and another one will be started. So, but everything starts from the primer, which is not used up in starvation. That primer always stays there. All right, great. Got ahead of my slides a little. Each glycogen contains 4 enzymes. So the enzymes aren't drawn in here except the glycogenin, which is the core. So glycogenin is the core of the glycogen molecule. It contains a primer that was built by the glycogen. It contains a really important enzyme, glycogen synthase, glycogen phosphorylase, branching and debranching enzymes. So those are all within the glycogen unit or the glycogen molecule. Let's build a glycogen. So to convert glucose 6-phosphate into glucose that you can stick on a branch, like we're going to be talking about sticking one new, I guess they're all branch points here, but we're going to talk about how to stick one of these on. Actually, we're going to talk about sticking the very first one on a primer is what we're going to do. But yeah, there's 6 enzymes. You can read them here. We're going to go through them though. So let's not go through them right now. So here's the story in a nutshell. This is glycogen synthesis, also known as glycogenesis. And so we've already made glucose 6-phosphate, but we can do it again. So glucose comes into the hepatocyte. It gets in through a GLUT2 transporter, and it's immediately converted into glucose 6-phosphate. The hexokinase is the one that converts it into glucose 6-phosphate. In the liver, it's glucokinase. In muscle, it's hexokinase. That's actually hexokinase 2, I believe. Remember, the AKA for glucokinase is glucokinase 4. But anyway, this is the path we're going to go through. So let's just go through it. Glucose 6-phosphate then is converted to glucose 1-phosphate. Okay? And then the glucose 1-phosphate is converted into the important one. It's called UDP glucose. So glucose 6-phosphate morphs into glucose 1-phosphate. There's the enzyme. It's phosphoglucomutase is the enzyme that does that. If it's not on here, I'm not going to hold you responsible. I'm more interested in these enzymes, these trapping enzymes, and the rate-limiting step. So glucose 6-phosphate is converted to glucose 1-phosphate. And that is converted into the all-important UDP glucose. So 2 enzymes are needed to do that step, so I wasn't that excited about that. Then, to activate glycogen synthase we'll learn is the one that actually sticks the glucose to a branch. It's normally not activated. It's full of phosphates, and the phosphates deactivate it. So we can use the energy of UDP molecule to rip or to pull the phosphate out and stick the glucose on there. I just think of that as like a little power pack or a booster. If we break that off, it's going to give the glucose enough energy to bind on the branch and become part of the glycogen molecule. So let's see what I wrote. The active form of glycogen synthase grabs UDP glucose and it pops off the UDP group, and that gives it the energy to stick that glucose on a branch of the glycogen granule, or just on the branch of the glycogen is fine. So glycogen synthase is the star here. And we can see number 5 is glycogen synthase, and there's some fun facts about it. It's the rate-limiting step of glycolysis, or rate-limiting step of glycogenesis, or glycogen synthesis. So I always ask that, and boards always ask that. So definitely know that one. All right. Also interested here, glycogen synthase is stimulated into action by high levels of insulin, and we're going to get into the weeds on that right now. Let's go a little bit deeper and check out this glycogen synthase, because that's the star of the show. That's what actually sticks the glucose on the glycogen, the growing glycogen molecule. So glycogen synthase is in 2 forms. Normally it's inactive, and it's inactive when it has a lot of phosphates on it. And who puts those phosphates on there is this glycogen synthase kinase 3, or GSK3 is the one that usually does that. So in order to wake this up, we need to do a couple things. So notice insulin is up here. You can think of this as a hepatocyte. Insulin binds, it can't go through the cell membrane, so it binds to its tyrosine kinase insulin receptor, which is on the cell membrane. And that triggers a pretty famous second messenger system. And that's called the PI3K AKT pathway, where AKT is the actual effector that does all this stuff. And so the AKT with insulin will inhibit through this. AKT will go and turn off this glycogen synthase kinase. Normally this is on by default, but if you turn that off, then you can't put any more phosphates on there. So that's part of the story. So that one's off, but now we got to activate this because it's still deactivated. So to activate it, you can also see insulin also binds to the same hepatocyte, and through that same PI3K AKT pathway, the AKT stimulates a important protein here called important enzyme called protein phosphatase 1, or PP1. And PP1 will go up here, it'll bind to the glycogen synthesis, and it'll rip all these phosphates off. And so you can see we go from the inactive form, the active form, or the inactive form of glycogen synthase is called glycogen synthase B. So if PP1 is activated by insulin, and if GSK3 is inactivated also by insulin, then PP1 will wake this guy up by pulling off its phosphates, and now we have the active form of glycogen synthase, which is called glycogen synthase alpha for active. See how that works. So everything I said in these slides, so we'll just read through them again. The key enzyme that makes or puts glucose on glycogen is glycogen synthase, specifically the activated form, glycogen synthase alpha. So glycogen synthase is the rate-limiting step of glycogen synthase, of glycogen synthesis. And yeah, the activation of glycogen synthase is started when insulin levels are high. Yep, because we need insulin. We need insulin to turn off the inhibitor here, GSK3, and we need insulin to wake up PP1. PP1 will then bind to this, knock off those phosphates. Once these phosphates are off, we have an active form. And now glycogen synthase will go and it'll take that UDP glucose, where is it? UDP glucose, and it'll stick it on, and we'll look how that works here in a second. It'll stick it on one of the branches of the glycogen. Okay, so hopefully that sinks in. Just go ahead and listen to it again if you have to, and then again if you have to, and everything we said. So dephosphorylation is accomplished by who? Protein phosphatase 1. I think of phosphatases as like tasers. They're going to hit the target and they're going to tase it, and like in Star Trek, and knock off phosphates. They don't kinases put phosphates on. These tasers, phosphatasers, take phosphates off. So dephosphorylation is accomplished by the enzyme protein phosphatase 1. It is insulin and glucagon sensitive. And we might as well look at glucagon real quick. So if glucagon's around, insulin is not around. Remember, those two are not around. And it's one of its favorite helpers is epinephrine here. So they actually, through their own second messenger systems, which we won't worry about, they make sure that PP1 stays inhibited. And if it stays inhibited, then you're going to stay in this inactive form. And we certainly don't want this pathway running if there's no blood sugar around. So glucagon will only be out when there's no blood sugar. See how that works? So everything we said, insulin binds to its receptor in hepatocytes, triggers the PI3K ART pathway, and that activates our friend PP1. PP1 then dephosphorylates the glycogen synthesis beta, which is the inactive form, goes over here, takes these phosphates off, and that becomes activated. Cool. Remember, insulin does not directly activate anything, right? Insulin can't go inside the cell, right? It's a peptide hormone, so it can't go through a phospholipid bilayer like cortisol and aldosterone can. So therefore, it has to use a second messenger system. Great. Note in the liver, PP1 is activated, stimulated by increased levels of insulin, as well as increased levels of glucose 6-phosphate. So this whole, and that's the starting molecule of this whole pathway, so it can kind of push itself if there's a lot of glucose 6-phosphate around. Note, insulin also inhibits GSK3. We talked about that already. GSK3 normally inhibits glycogen synthase by phosphorylating it, right? Because it's a kinase. Kinase phosphorylates. And now you got to be careful, because sometimes when you phosphorylate something, it activates it. And sometimes when you phosphorylate something, it deactivates it, which we're going to see in a little bit here. All right, fun fact down here. Remember, hexokinase 4 or glucokinase is not allosterically inhibited by glucose 6-phosphate. Therefore, hepatocytes can accumulate a lot of glucose 6-phosphate, which can then drive into glycogen synthesis and de novo fatty acid synthesis, and pentose phosphate system as well. All right, here's just a little cartoon, just a review of how this works. So there's a glucose. It gets trapped by glucokinase. This is a liver cell. The glucokinase sticks a phosphate on there, so we got glucose 6-phosphate. Go through a couple steps and make UDP glucose, and UDP glucose is the one. It doesn't by itself. It's UDP glucose is grabbed by glycogen synthase A, the active form of it, and it doesn't need ATP to do this. It uses the power to make this reaction. It uses the UDP, rips off the UDP part, and then the glucose is just stuck on the available branch of the glycogen molecule. Another fun fact here, when the first primer branch gets too long, or when any branch gets too long for that matter, you can't get more than 11, 12 units or glucose units per branch. Then an enzyme called branching enzyme will come and cut the extra glucose monomers off that branch and reattach them to that chain. So bottom line, it doesn't let these chains get too long. It keeps trimming them and making new chains. This here's kind of a cartoon of a glycogen molecule. Yeah, glycogen is a giant molecule found in the liver. We know this, made of thousands, 55,000 was it? Glucose monomers hooked together. Its formation is driven by insulin and glucose 6-phosphate, which stimulate glycogen synthase basically. During fasting or under the influence of glucagon, these glucose monomers from the glycogen tree can be cut off and released into the blood as free glucose. And this liver glycogen lasts for 12, 16, 18, up to 24 hours. We're going to go with 24 because it's easy to remember. But if we see a board question, it says 16, 18, or 24. That's how long glycogen can be used to make glucose or put glucose in the blood. After that, we have to go into gluconeogenesis. Fun fact, kind of said this already, 1 molecule of glycogen can hold 55,000 molecules of glucose. There's millions of glycogen molecules in the liver itself. During fasting, liver glycogen only lasts for 24 hours. We said all this already. Skeletal muscle glycogen can be used by skeletal muscle In other words, skeletal muscle glycogen can be broken down into glucose, so skeletal muscle can run that and make energy through ATP during fasting or whenever. But it's selfish. It can't release it to service the brain and red blood cells and such. How come? Because this is definitely on the test. How come skeletal muscle has all this machinery, but it can't release? It can get to a glucose 6-phosphate. It can break down a glycogen molecule down to glucose 6-phosphate, and that's it. It can't make glucose. Why? Because it doesn't have glucose 6-phosphatase. Right? Only liver, only hepatocytes. So glycogenolysis. So now that we built the glycogen molecules, it's like 2 in the morning and you definitely have run out of blood glucose. And so the first thing that happens is glucagon is released from the pancreas. Insulin goes away, glucagon is released. And glucagon will stimulate the breakdown of these glycogen molecules. In other words, glycogenolysis. Lysis means breakdown, and that's sugar. So breakdown, or that's glycogen, right? Glycogen breakdown. All right, everything I said, about 4 hours after a meal, all the blood glucose is gone. So we learned that certain cells, red blood cells, brain cells, renal, not the adrenal medulla, the renal medulla must have at least some blood glucose. So what's the solution? It's time to tap into those glycogen molecules and those glycogen reserves. And it's time to run glycogenolysis. Now, gluconeogenesis is also starting to ramp up. But that system doesn't really start producing tons of glucose until after about 24 hours after the system is exhausted. But these two systems are running concurrently. And yeah, glycogenolysis to the rescue. Remember the glycogen molecule we just built in the liver? It's time to rob some of that glucose and put it in the blood to save the day, to feed the red blood cells and brain. So what's the first step? How do we break glucose off from the glucagon molecule? Well, we already said this, but glucagon is released, and glucagon will bind to hepatocytes, and it has a GPCR, glucagon receptor is what it binds to. It's a peptide hormone as well, like insulin, so it can't go inside the cell. So it's going to work with a second messenger system. And this one is the classic number one, probably the first one you ever learned. This is the cyclic AMP PKA, protein kinase A. And PKA, I'm just going to call it from now, that's the effector. That's the one who does the dirty work. So now we're going to talk about PKA and how it works. So what are the actions of PKA? So, and here's the deal. So there's glycogen synthase that we talked about. So we're not going to be doing this, right? We're not going to be adding glucose to the glycogen molecule because there's no blood glucose around. So this needs to be shut down, and that's exactly what happens. So glucagon binds to the hepatocyte cell membrane. It triggers the cyclic AMP PKA second messenger system. PKA will go and it will inhibit glycogen synthase by also adding phosphates to it. It's just another mechanism to shut that thing down. And it just shows in here that, remember, when insulin on a fed state, when there's lots of blood glucose and we want to store some for a rainy day, then we use this PI3K AKT pathway, and that's going to stimulate PP1, and PP1 is going to remove those phosphates and activate. We just talked about that. But right now, no insulin, so this is off. So glycogen synthase is inhibited, and we're going to go the other way. Now we want to take the glycogen and pull glucose monomers off it and get those things in the bloodstream. So what we can do with this step is make glucose 1-phosphate. And so there's another enzyme called glycogen phosphorylase. I don't think we said that yet. That's the key. That's the the opposite twin of glycogen synthase. Glycogen synthase makes glycogen. Glycogen phosphorylate breaks down glycogen, steals the glucose monomers. So how does that one work? Well, here comes our glucagon, and it stimulates the cyclic AMP pKa second messenger system. pKa is loose, but pKa can't bind to glycogen phosphorylase directly, like pKa here can bind directly, or pKa can bind directly to glycogen synthase and knock it out of action. Not true here. We need an intermediate. And so the intermediate is called phosphorylase kinase. So pKa binds to phosphorylase kinase and wakes that up, and that can phosphorylate glycogen phosphorylase into action. Remember, if you phosphorylate glycogen synthase, it deactivates it. But if you see the add phosphates here, that's phosphorylate. So if you add phosphate groups to glycogen phosphorylate, it wakes up and it does its thing, and it pulls, and we'll see on the next slides, but it grabs a monomer, a glucose monomer, it shoots a phosphate at it, and it rips it off by shooting the phosphate at it, and it makes a glucose 1-phosphate. Okay, and just on the other side of the coin, there's insulin. I think we talked about that. There's PP1, and we talked about how that gets inhibited by adding phosphates, which is really strange. All right. Fun fact, pKa always phosphorylates. Note, glucogen-driven pKa cannot directly activate glycogen phosphorylase. It must use phosphorylase kinase, a middleman, which actually goes out and gets the job done, which activates glycogen phosphorylase via phosphorylation. And that's actually, did I say, that removes phosphates, PP1's effect on glycogen phosphorylase. PP1's effect on glycogen synthase is it to make it active? Yeah, it removes them here as well. So PP1 always removes phosphates, but it has, again, different functions on both of these different enzymes, which is pretty crazy. Yeah, so once we activate glycogen phosphorylase, it attacks, and you should know this, it attacks the alpha-1,4 glycosidic linkages. What the heck does that mean? Well, here's a glucose molecule. Remember how they're labeled. You always put the, when you lay out a glucose molecule, 6-carbon molecule, you always put the O up at like 1 o'clock. And then that'll be number 1 carbon. And do we know how to read these? Hopefully. These carbons aren't drawn, but they're the points here where these two lines come together. So there's carbon 1, 2, 3, 4, 5, and then the 6 is up in the air. So on this one, there's the 1. So this is where an alpha-1,4 glycosidic linkage occurs. And what exactly happens? Glycogen phosphorylase will throw a phosphate basically at this thing, and it'll stick at this 1 carbon here, right? And there's the throwing of the phosphate, and you end up, look at that, we got the phosphate stuck to the number 1, or this would be the last glucose monomer on a chain, and the chain would have 6 more glucoses here, but this is the very last one is what gets attacked. And how do you attack it? Glycogen phosphorylase throws a phosphate at it, and it breaks this bond. This phosphate hits the bond, and it sticks. And you can see it stuck here now. Sticks to the oxygen. And then just a hydrogen is, and an OH is left over here. We won't worry about what happens there. But yeah, and so great. And now we got, what do we have? We have glucose 1-phosphate. And that's all the farther we have got. Now we just have to morph that back into glucose 6-phosphate, which we'll do next. So see how that works. It's not too bad, I don't think, if you study this and look at it a little bit. So another key about glycogen phosphorylase here, it is the rate limiting step. It is the rate limiting step. Great. So now we got glucose 1-phosphate. Now we just have to convert it to glucose 6-phosphate, and there's an enzyme called phosphoglucomutase. Phosphoglucomutase takes that, and it's a two-way reaction. It can go both ways, because remember, when we're making glycogen, we went this way with it. Now we're going the other way. It's the same enzyme. So phosphoglucomutase. Great. That's not stimulated by glycogen. Another one stimulated by glycogen is the main taser here, glucose 6-phosphatase. This is one that muscle doesn't have. Only liver cells have that. And so that will happen next. Once glucose 6-phosphate is around and glycogen is around, glycogen can bind kind of indirectly to both of these through pKa, but it stimulates this into action. It grabs glucose 6-phosphate, rips the phosphate out, and you got good old glucose. And out you go through a GLUT2 transporter once again. And therefore, the red blood cells are happy because they got some fresh glucose coming in. And plenty of glucose to go around. It'll take it to the brain. It'll take it into the renal medulla. And everybody will be happy. Yeah, everything I said. Glucose 6-phosphates convert back into glucose by glucose 6-phosphatase. Glucose 6-phosphatase is also strongly stimulated by glycogen, just like this picture shows. And the newly made glucose is then released from the hepatocyte into the blood and feeds the tissue that needs to be fed. And just a note about, so that's it. Really, we're done. So it wasn't that bad, I don't think. Just a note again about skeletal muscle. We already said this, but skeletal muscle, they can run glycogenesis during the fed state and store glycogen. And then they can do glycogenolysis, but they can't release it to the body. They get stuck at glucose 6-phosphate. And the glucose 6-phosphate will just run through glycolysis and then through the Krebs cycle and then into the electron transport chain. How come? Because skeletal muscle cells do not have the taser, the main taser, glucose 6-phosphatase. Okay, so hopefully, I mean, you've had this all before, right? So this should be just a simple review for you. So if you've never had this before, I could see how this would be tough for you, but you've had it before. You've had 2 biochem classes, so I'm confident you'll get this stuff down. Email me questions. And the Northern Harrier says, see you later.